Thank you very much for your patience in getting this set up and to the organizers for this great opportunity to present our work today. So as Edith said, I'm associated with the Odom lab at DKFZ and the Heard lab at EMBL, and my project is about exon activation, and not only in the context of the adult mouse liver. As already very nicely introduced by Daniel earlier today, sexual dimorphism in shape and size and also behavior is widespread across animals. But what might come as a surprise to some of you is if you zoom in a little bit deeper and look, especially with the latest molecular technologies, at just about every organ in the body, you will find a sex difference. And in this project, we focus on the liver for the following reasons. I mean, to cut a long story short, it's super different between the sexes in just about every way. We came to this topic through the lens of cancer. I'm at a cancer institution. We wanted to understand why males are around 3 times more predisposed to liver cancer than females. But then we realized pretty quickly that the story extends way beyond cancer. And so the working hypothesis, and also the reason why we do a lot of our work in young, healthy tissue these days, is that sex differences in healthy tissue or healthy hepatocyte physiology in a non-cancer context actually predisposed to cancer later in life. And so, of course, there's been a lot of work done on sex differences in cancer. I mean, cancer is not an uncommon disease. There's been a lot of work done on extrinsic factors. But I think the fact that we see this cancer sex bias, or this liver cancer sex bias, also in mice that are kept under the sex-invariant environmental conditions, strongly suggests a contribution of intrinsic factors as well. And in particular, gonadal hormones, so testosterone and estradiol, and also sex chromosome complement, so XY in males and active X, inactive X in females. But as Igneza already very nicely introduced earlier today, the inactive X is not completely switched off. And in every context that's been looked at, there's a subset of genes that are to some extent expressed. I would argue it's usually not quite a double dose, but certainly an appreciable signal. And so the hypothesis that we developed 3.5 years ago is that these SKPs might be at least part of the reason why male humans and mice are more likely to get liver cancer. And so to test our hypothesis, we have 2 big directions. And because I only have 10 minutes, I'm just going to briefly touch on both of these, but would be very happy to discuss later. So first, we are looking very carefully at the inactive X in single cells in single adult mouse livers. Which is a complex problem, as I'll get on in a second. And then second, we're zooming out a little bit from the inactive X. And as I alluded to, over time, this not so inactive X focus has become a rather big part of our program to look at the contribution of sex chromosome complement and or gonadal hormones in this complex phenotype. Remember, this is a liver in a mouse with a bloodstream and a lot of things going on. So of course, it's not only going to be one or the other. And so just first to the first point. So the general approach that we take, and this is an approach set up with 3 wonderful collaborators in the Odom lab a couple of years ago now, is allele-specific single nucleus RNA sequencing. And so why do we have to do this mouthful? Well, as I alluded to, a wild-type tissue in a mouse is not a controlled system where we can reproducibly always inactivate the same X chromosome. It's a mix. And when we do this in an F1 hybrid mouse, it's not exactly 50-50, as many of you know, there's a skewing. But you still have very large populations of cells that silence one way and very large population of cells that silence the other way. And so you really do need single cell resolution if you want to do this in a complex tissue. In addition, liver, hepatocytes have some other quirks like polyploidy that I'm not going to talk about today, but single cell resolution is a very useful thing. And so after trying lots of different approaches over the last 3 years, I'm just going to briefly flash up the one we've converged on now. And so we isolate nuclei from frozen tissue, and we always keep the nuclei separate with respect to which mouse it came from. That's very important. We can do this with very low amounts of material, which means we can get error bars on our escape plots and see how individual variability is. And the library preparation approach that we use is skyRNA sequencing. If you click on the QR code, you'll get a link to our updated version of the protocol for adult tissue. But it doesn't end at the bench, and in fact, this is the reason why it's taken 3 years to get to this slide. We had to develop, or Yob had to develop new tools to haplotype the reads. So we're able to assign about 30% of our short 3' reads to one haplotype or the other, so Castaneous or Black 6 in this case. And then once we have these data, actually do the allele-specific analysis using a new package called SkyR, which you can also find here. And so in one of our recent data sets, we had around 500,000 cells. This is a UMAP representation. Long story short, individual points represent individual nuclei, and how close they are together approximately resembles shows their transcriptional resemblance. And in this data set, we have around 150,000 female hepatocyte nuclei. So all of the data I showed today will be in hepatocytes, but we can discuss other cell types later if you're interested. And if we zoom in on these 190,000 nuclei, and we plot the amount of signal from genome 1, cast, versus genome 2, black 6, on these two axes, and each point represents an individual nuclei, and we really have like more than 100,000 nuclei here. They're very, very, very densely packed. What I hope you can appreciate is that the vast majority of the nuclei fall here in blue. These are ones where at least 90% of the signal has been assigned to the black 6 genome, and therefore we assume that it's the Castaneous inactive assumption. And then also the other way around. And what many of the ex-aficionados have probably noticed already is that there's a pretty strong skewing here, but not necessarily in the expected direction for this cross. We've double, triple checked our code. There doesn't seem to be a technical explanation. We're looking into it. And there's also around 18% of nuclei in the middle here where we cannot assign an XI unambiguously. These could be doublets, though we are taking various steps to prevent that. My gut feeling is that these are polyploid nuclei. Liver nuclei can have 16 or 32 X chromosomes, and we for sure have a population of those here. We're a little bit blind to it, and so it may be that we don't always have this strict following the rules of, always the same species as you scale up. But at any rate, 82% of the nuclei, with a few assumptions, we can assign an XI, which means we can start to ask about which genes are expressed from the inactive X. So in wild type, healthy liver tissue, these are 10-month-old mice in this case, because they're controls for a group I'll show you in a second. I'm showing you escape from the XI quantified here. So on the y-axis is the allelic ratio, XI reads over total. And then on the x-axis is our proxy for kind of total expression. This is summing up all of the XA and XI assigned counts. And the points that are labeled are the points with an allelic ratio of above 0.1. So first, as a sanity check, it's nice to see that exist is throwing an allelic ratio of 1, even in these hepatocytes where we could have very funky things going on with ploidy, as I said. So that was nice to see. And second, the vast majority of the genes are falling pretty close to 0. Not exactly on 0, and I think there is some technical contribution here as well. But by and large, I think we can take it at face value. And what I guess isn't coming as a surprise to the XCI aficionados in the audience is the usual suspects are popping up. Very important genes, for example, KDM6A, 5C, and DDX3X are well-known, well-characterized tumor suppressor genes. So yes, the same constitutive escapees, but potentially with a very important contribution here. Beyond those, when we start to get into the 0.07 allelic ratio, I say these are maybes. And follow-up and validation is really required. So this was cool to see, though it doesn't look like there's a whole lot of at least very strong escape in adult hepatocytes, if I may, which was on one hand a little bit of a disappointment, but also really interesting if you think about it. And now if we take 7 mice that were fed a really nasty diet for 10 months, exactly in parallel as these guys, and everything done at the same time, you see an extremely similar picture, which is, again, very interesting because on this diet, if you wait 1 or 2 more months, the males will all get liver cancer, whereas the females will only 1/3 of the time get liver cancer. And yet, despite of that really nasty carcinogenic stimulus, putative escape seems to be largely unaffected. And if we take the exact same data and we plot for each of the 9 putative escapees at individual resolution, where each point represents an individual liver, what I hope you can appreciate is there's some variability, which is very interesting, also then possibly comparing that to other things that we measure, but that it's rather stable. Okay, so clearly there's some escape happening in the liver, I think it's fair to say, but not a whole lot, which is also why, in parallel with the work I just showed you, we were exploring a little bit more broadly. And so now just to give you a taste of what we're doing there. So we use the 4 core genotypes mouse model, which, to cut a long story short, generates litter-mate mice of 4 different groups, which allows for comparisons of sex chromosome complement and gonadal hormones in vivo and whatever your organ of interest is. And so the experiment we set up here is to collect 48 livers from all of these different groups and do paired single nucleus RNA sequencing and lipidomics. And then for each of these readouts, we asked, what is the contribution of a sex chromosome complement really, in this complex in vivo context to physiology? So zooming in on 300,000 hepatocytes now, if you compare XX and XY, you find several very important genes, Y-linked genes, and genes associated somehow with X chromosome inactivation, which was very nice to see as a sanity check, but not exactly consistent with a really large contribution of sex chromosome complement to liver physiology. Conversely, if you compare with respect to gonad, you see more than 1,000 differentially expressed genes. They're very densely packed in there, including many known sex-biased genes, but also many new ones. So at the level of gene expression, it looks like sex chromosome complement has a rather mild contribution to sex-biased physiology, whereas hormones are a relatively major one. And so then on the other hand, we looked at the exact same 48 liver samples at lipid abundance. Liver is a very important organ with respect to lipid metabolism. Comparing XX and XY, we actually don't find a single significantly different lipid, and we have about 1,000 lipids in our data set here. But when we compare with respect to gonads, very large effects again. So also at the level of lipid abundance, sex chromosome complement does not obviously impact sexually dimorphic tissue lipid levels. Yeah. Okay, so just ending now, because I'm basically out of time. After establishing that it seems like there's quite a strong contribution of gonadal hormones to sex-biased liver physiology, we of course also want to understand the mechanism. So the way we do this is we team up with computational folks like Aurelian that actually know how to do this stuff rigorously. He uses tools like Morpha to basically take our RNA and lipid data, feed it all into an algorithm, and then ask which genes and lipids are behaving in similar ways. And then he builds a very simple, as he says, correlation network, which is quite a mess. I won't get into the specifics now. But what I hope you can appreciate is there's 4 genes in the middle, these 4 CYPs, that are connected to lots of different lipids. 4 genes, nice. That's a number we can work with in the lab. And so I've started to do functional screenings now in vitro and primary hepatocytes. And this is my last slide. And so we've now done this experiment 3 times. We see a phenotype for several of our genes, which was cool. But for one gene in particular, we see a relatively strong phenotype. So I wanted to show you. It's work in progress. So this is CYP2C38. It's relatively lowly expressed. That's why the control levels by RNA-seq are quite variable, but I hope you can appreciate we can knock it down. And when we do, here plotting the in vitro knockdown effect size in primary hepatocytes on the Y and the in vivo correlation on the X, we find that many of the lipids in our data set are not corresponding in the way that would be predicted, but that there is a subset shown here in the top left and here in the bottom right that are including some very significant effects. And what's particularly interesting about CYP2C38 is it's not equally expressed everywhere in the liver. It's highly expressed in about half of the hepatocytes. These are the CYP2E1 positive hepatocytes shown here by spatial transcriptomics, and here I show you the CYP2C38 expression. So we think that CYP2C38 is a key regulator of sex-specific lipid levels, and not equally throughout the liver tissue, but preferentially in this pericentral region, which has very interesting implications for liver biology. Okay, I'm not going to read all of the summary now, I'm out of time. But just to get to the most important slide, really big shout out to my 2 mentors, Duncan, who's here, and Edith, who's of course also here for constant support, even with this project that's taken us in all kinds of directions. Direct contributors to the work I've shown today, the Heard Lab for welcoming someone who isn't around so much, but they're always very helpful, and the Sander Stiftung for funding the work, and you for your attention.